Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Jean-Pierre Roussaire, Assistant Professor, Boston University School of Medicine. Jean-Pierre is also a 2023 Toffler Scholar. Today's discussion is focused on deciphering the molecular events leading to neurodegeneration in Alzheimer's disease. So let's dive in. So welcome, Jean-Pierre.
Thank you so much, Deb. I'm very excited to be here talking with you about my research.
Yeah, I'm excited too. I love your passion for what you do, and so I'm very excited for this discussion. I would like to just start out. You've got a pretty interesting academic journey so far. You know, you started with engineering and then you moved to neurovirology. What inspired you to transition? And especially from neurovirology, what is it? Explain what it is. But now you're focused on Alzheimer's disease research. Can you walk us through this, these steps?
Sure. So I was doing my PhD at the Pasteur Institute in Paris, and I was studying a virus that can travel from the brain of a mouse into its spinal cord and stay for the entire lifetime of the mouse in the spinal cord. And it's kind of a decent model for multiple sclerosis in the mouse. And I was studying how the virus was traveling from one type of cell to another within the central nervous system of the mouse. And so obviously, because the virus travels through the mouse brain, I was studying the central nervous system. I was studying neurons, and I was studying the supporting cells that are around neurons. And I thought that this was also exciting. But this was just the playground of the virus. This virus is a virus that only infects mice and not humans. And so I was a little bit frustrated, first because I wasn't studying a major public health problem. And I was also frustrated because I was only studying everything from the perspective of the virus and not from the perspective of the healthy brain. So it was not so much about understanding how neurons were functioning, how the brain was working in order to deliver cognition and memory. It was really how can the virus divert the function of the brain in order to establish its infection. So after having spent quite a few years working on that, I really wanted to focus more on a real human public health problem, and also studying more the basal function of the brain and not in the context of a viral infection. And so this is what led me to the United States, where I did my postdoctoral studies at Rockefeller University, and I completely forgot about viruses, and I started to work on Alzheimer's disease. And this is basically the roughly the same project that I've been working on for now almost 2 decades.
Wow, thank you. You know, there's I've read some things that some have maybe hypothesized that Alzheimer's could be a virus. Do you have any thoughts on that?
Yeah, this is a very interesting hypothesis. I have not myself done any experiments that could conclude one way or the other. I think that there's very interesting evidence in the literature to support that. I still think that we're pretty far from being able to ascertain that Alzheimer's is coming from a virus. And I think that there's reasons to think that maybe not. But my research is definitely not going in this direction.
Yeah, and I didn't mean to put you on the spot, but I just thought it was kind of interesting, your background. So your lab, because you've established this lab, and its focus is on deciphering these molecular events that really is kind of leading to this degeneration of cells in Alzheimer's disease. Can you share some insights into, just kind of the day-to-day lab, who's in it, what you're working on, and any unique approaches that you are using to kind of create this new knowledge and understanding that is so important.
Sure, of course. So it's I started at the lab a little bit more than a couple of years ago, and everything is taking time in research. So the lab is growing. I have 3 graduate students, a research assistant that are working with me. We have undergraduate students that rotate in the lab. So the lab is growing little by little, and we can tackle more and more different directions. But we are characterized by a pretty intense focus on one very, very specific question that I think holds major promise for mechanistic insight into the disease and for new therapeutic avenues. And this focus is a focus on the very first neurons that are affected in the brain of people with Alzheimer's disease. It's neurons in a region of the brain that's called the entorhinal cortex. And even within the entorhinal cortex, it's specifically neurons that are in a part of the entorhinal cortex that's called the layer 2 of the entorhinal cortex. So these neurons in the layer 2 of the entorhinal cortex, they are forming protein aggregates of a protein that's called tau before any other neuron of the brain. And they are degenerating before any other neuron of the brain. I should say not every Alzheimer's disease patient, but every patient that has a classical form of Alzheimer's disease. So a classical amnestic form of Alzheimer's disease where you lose the ability to form new memories. And our reasoning is that if the vulnerability of these cells is so robust that in roughly every patient that has amnestic Alzheimer's disease, these neurons are hit first, we think that there's a lot of things that we can learn if we understand the particularities of these neurons, if we understand what's going on in these neurons at very, very early stages of a disease, and if we understand why it's not other neurons that are the first ones to be affected. And these neurons are really, really cool. I mean, we're so intensely focused on them that I've grown a very special relationship to them. They are super important for new memory formation. They are the neurons that root inside of a brain structure called the hippocampus. They're rooting inside of the hippocampus all of the sensory information that your brain is processing as you're evolving in the universe. And so they are responsible for bringing into the same place all of these sensory information to assemble new memories. And so probably they have to be very plastic because they have to constantly form new association between sensory information. And they're also absolutely indispensable for new memory formation. So when these neurons go, your brain is unable to form new memories. And this is what's happening in patients with Alzheimer's. And so we are trying to develop the tools to study these neurons, trying to re-examine a lot of things that have been done in the field of Alzheimer's disease from the vantage point of these neurons, and trying to be really, really rigorous in studying these neurons and not other neurons. So to give you an example, a lot of Alzheimer's disease research needs the ability to model neurons in vitro, so to grow neurons in a dish, to be able to we maintain them easily for weeks or a month or a few months in an incubator. And they are recipes for making just generic neurons that are not necessarily going to look like our favorite neurons in the entorhinal cortex layer 2. And these neurons have been heavily used by the Alzheimer's disease community. And it's great that they were used because it was the only tool that was available. But we think that studying these very generic neurons that don't look quite like these entorhinal cortex layer 2 neurons is maybe not going to allow us to completely understand all the steps that are leading to degeneration in the Alzheimer's brain. We think that it's actually really, really important to try to make these neurons in vitro. And so this is one of the things that we're doing in the lab to try to find the recipe to make these neurons in vitro. And we have some good clues, and we're starting to do experiments with these neurons. But this is one of the ways in which we think it's really, really important to be very precise, and that what we're studying is actually the neurons that are the very first ones to be sick in Alzheimer's.
So this may be a wacky question, but if these neurons are that special about our memories and kind of our cognitive behavior, understanding these neurons, would this also give us insight into what cognition really is?
Yeah, that's a really great question, and very much so. So I think this is always this. I always like to think of pathology and of disease as just a subversion of physiological mechanisms. It's not like there's that the brain has mechanisms of disease that are special and that don't have anything to do with the way it's functioning in normal times. And I think that the reason why these neurons are very vulnerable in the case of Alzheimer's disease, it's because they are doing something particular for memory formation. And I think it probably has to do, and this is not demonstrated, but I think that it has to do with the fact that they are so plastic that they constantly need to reshape themselves to make new connections with other neurons. And this comes at a price. Having a structure that is so plastic that can remodel itself so much potentially is making that structure a little bit more fragile. But I think that once we understand the particularities of these neurons, we'll be able to understand new things about memory and new things about Alzheimer's disease.
Is it a giant leap to say that if you can develop these neurons in the lab and you can create them, that you could actually implant them then if people have that have the disease and those neurons are dying?
That's such a great question. This is something that I think a lot of people in neurodegenerative diseases, but not just in any disease where you have a type of cell that is dying, the idea that you could potentially make these cells in vitro in a dish and then implant them. And for a lot of things, actually, Parkinson's disease research is a little bit ahead of Alzheimer's disease research. And in Parkinson's disease, you have a similar vulnerability of a particular type of neurons. It's the dopaminergic neurons. And people have been able to do for a while dopaminergic neurons in a dish and have tried to implant it, not exactly in the region where they die, but in another region where they are actually very useful. And actually, there were some promising results. There's also a lot of complications that occur, but there's some promising results. The only difficult thing with our favorite neurons from the layer 2 of the entorhinal cortex is that they have the most beautiful, most complex axonal arbors, so axons. So it's the axon is this process that's coming out of the neuron and that the neuron uses in order to talk with other neurons. And the axons from entorhinal cortex layer 2 neurons are extremely ramified, extremely complex, and I doubt that we'll ever be able to implant a cell in the human brain that is going to be able to grow such an amazing axonal arbor like the original entorhinal cortex layer 2 neuron has. So I think it's possible that we'll be able at one point to find a way to at least replace some of the function of the dying entorhinal cortex layer 2 neurons, but I think that it's definitely going to be a very challenging task.
And these axons are, if people have seen the neurons, it's like the little arms that come off of the main body, and some of them have almost like little scoops or something at the end of them, and that's where they're interacting with other things in the body. Is that the picture you're trying to paint there?
Exactly. Except that so in general, on a textbook drawing of a neuron, one of the types of arms that the neuron is growing, the dendrites, are very complex and ramified. But the axon always comes up for some reason as just a one arm that's simple and long. The axon is really the longest arm that's coming out of the neuron. And actually, for a neuron like the neurons from the entorhinal cortex layer 2, this could not be further away from reality. It starts as one arm, but then it branches and it forms just a forest of arms that are conveying all of the electrical signals that are coming from entorhinal cortex layer 2 neurons.
Well, that makes sense. If it's if they are the drivers of all of our cognition, and it makes sense that they would have multiple inlets.
Yes, specifically of the memory part of the cognition. Yes, absolutely. Yeah.
So I'm curious, your engineering, undergraduate degree, how, and you're talking about technologies that you need in the lab. Is there any relationship there from your distant past in what you're doing right now? Is that helpful to you?
That's a great question. I would love to be able to say that I'm using my engineering background a lot. I don't think so. But I think that the way this has really influenced me a little bit is that I'm really drawn to more quantitative approaches. And sometimes biology can be a little bit descriptive and not be too quantitative. But from the beginning on, when I started to try to address this problem of the vulnerability of these neurons, I was wondering how we could potentially come up with that kind of big data driven approach to that. And so now for the past 10 years, I have collaborated extremely closely with a few functional genomicists. So these are computational biologists that are amazing at modeling tissues, modeling brain regions, modeling neurons, using genomics and genetics data. And so I was collaborating with Dr. Troyanskaya at Princeton University. I'm collaborating a lot with somebody who was a student in the Dr. Troyanskaya lab, who now has a lab at Rice University. And so with these computational biologists, we're working hand in hand in order to try to use all of the genomics and genetics data that we can in order to model computationally these neurons, and in order to kind of let the data speak by itself to point us towards the particularity of these neurons. And I think that if I didn't have this engineering background, so it's not like I'm the one that's actually, coding the algorithms, and they're doing that just so infinitely better than I could ever dream to do. But it helps to have a little bit of an understanding of what they're doing because of this engineering background. And so much of this exciting work with computational biologists is about designing really tools hand in hand that make sense for the biological problem that we're studying. And I think that there's no more fruitful collaboration than between an experimental and a computational biologist, because we have different ways of approaching the same problem. And I think only then can we manage to generate tools that are going to be really game-changing to understanding these neurons and to understand Alzheimer's disease.
Yeah, I think that's important. I had a previous conversation with Lydia McClure, who's actually has a background in science and research, and now she's CEO of an investment company. And we talked about, just the need for this community to understand the taxonomy or language of others, because whether it be researchers in business or biologists in big data, it's important because we need all types kind of work in this focus. When you're in the lab and you're trying to create the neuron, like what can you give us a picture of, like, how are you building these? What is it like? Is it
So we're not starting from scratch, and it's always like this in biology. We're building on something that has been done by somebody else to do something slightly different, and so there's a long history of people that have tried to make neurons from stem cells. So you can start from a stem cell, and by giving the right factors, small molecules, proteins, by giving the right factors to stem cells, you manage to instruct them to become neurons. And it's kind of recapitulating a little bit the developmental program that in the embryo is forming the brain from embryonic stem cells. And so there are protocols that exist, recipes that exist out there to start from a stem cell and to make neurons. But these are generic neurons. So neurons that are a little bit looking like all the neurons of your brain, but definitely not looking like our favorite neurons. And so we started from there. That was kind of our canvas that we wanted to, incorporate additional elements in order to make really what we wanted. And so we went back to our favorite computational biologists, and we asked them, can you, by looking at genomics data from these entorhinal cortex layer 2 neurons, can you identify elements that are very, very characteristic to these neurons, and that we could potentially try to put in stem cells in order to make them differentiate not into a generic neuron, but into our entorhinal cortex layer 2 neuron. And so we started to put some of these elements. We're still kind of refining our recipe and refining the different factors that we need to put in the mix in order to have a neuron that looks really like a neuron from the layer 2 of the entorhinal cortex. But we're definitely doing something that looks more like an entorhinal cortex layer 2 neuron than like a generic neuron.
I love that you have this real focus in your lab. Like this is what we're studying. What are the kind of the one or two major questions you're trying to answer? Because I imagine these questions up on the board, that's that focus. What are those questions?
So I would say the main overarching question is, can we describe, and this is basically the same question that everybody in the Alzheimer's field has, so we're not being original here, it's understanding what are the events inside of a neuron, the molecular event that lead to the formation of toxic protein aggregates, and that eventually leads these neurons to malfunction and to die. So that's the general question. But then we think that in order to answer this general question, we can use the biology of entorhinal cortex layer 2 neurons, because it's going to shine a spotlight on the features of a neuron that would make a neuron particularly vulnerable. And so our more specific question is, why is an entorhinal cortex layer 2 neuron so vulnerable? What is different between these neurons and other neurons? What is different between these neurons in humans compared to other species? Because humans are the only species where we see both the formation of these toxic protein aggregates and degeneration of these neurons. It's not the case in macaques. It's not the case in chimpanzees. It's definitely not the case in the mouse. So what is specific about these neurons in humans? And also in humans, at the very, very earliest stages of a disease, what are the molecular events that are occurring? We'd love to be able to nail down the very first things that are starting to dysfunction of these neurons, in these neurons. And so these are all questions that we're trying to tackle by using tissue that we obtain from, in particular, from the Boston University Alzheimer's Disease Research Center. So we obtain tissue from postmortem patients that died of either of Alzheimer's disease or that died of something else, but had some traces of Alzheimer's disease in their brain. We're obtaining tissue from macaques, from mice. So as I mentioned, we're doing all of these experiments in vitro with human neurons made from stem cells. And so all of these different models are helping us trying to address this question of what is the particularity of these neurons and why are they so vulnerable to the disease.
Is there any difference between these neurons in brains from different races? I know talking to people, our brain banks they're not homogeneous in just one aspect of, but they are primarily kind of US, European, primarily white. There's not a balance there of, people from South America and Africa and Asia. What's the hypothesis that you have? Could there be diversity in those brains considering these neurons, or do you think they're pretty common?
So I think that there's probably going to be diversity in these neurons between two individuals, and there's going to be as much diversity in these neurons between your neurons and my neurons and the neurons of just like anybody else. There might be some factors that are correlating with the ancestry, the complex ancestry that each individual has, that can explain the presence of a little bit more of this protein, a little bit less of that other protein. So definitely there are going to be differences. Some of them might map with the complex ancestry puzzle that is yours. But for sure, not every brain bank in the country, but so many brain banks in the country are very skewed towards the brain of white people. So unfortunately, we have a very narrow focus on one group of people. And there's a lot of brain banks that are trying to do a lot of effort to diversify a little bit their population so that we can start to ask these questions, not necessarily ask the question of what is the difference between these neurons between somebody of European ancestry and somebody of African ancestry, but more in order to accumulate knowledge on these neurons that are going to be valid for everybody out there and not just for people that are purely of European ancestry. So it's a really, really important question, and it's definitely something that I'd love to try to tackle a little bit more, to really make sure that we profile these neurons, that we make the inventory of all the proteins that are present in these neurons in a very diverse cohort of patients. That's for sure.
Yeah. Yeah, I've heard that, from many people, and it's just a robustness. It's a robustness to the research. It's and it seems like maybe there's something to be learned too about what may be going on in some people's brains that create protective mechanisms. That's kind of interesting. Yeah.
For sure. And actually, I mean, it's very, very premature. So a theory that I have is that when we compare, so I told you that mice in the mouse, these neurons were actually not very vulnerable. They're very vulnerable in humans. They're not very vulnerable in the mouse, and that's a significant hurdle for modeling the disease in the mouse, because we'd love to have a mouse model where these neurons are very sick like they are in humans, and we can try to prevent these neurons from dying. But unfortunately, in most mouse models of the disease, these neurons are living their best life until very late, while other neurons are dying, other neurons are forming toxic protein aggregates, etc. So I think that there are definitely protective factors that are present in the mouse version of these neurons. And so this is why I'm really excited about the comparison between species, because I think that we can potentially learn from these neurons in the mouse what are the recipe for being able to live your best life for very long. And potentially, we could, if we understand a little bit what makes a mouse neuron more resistant, we could make better mouse models that will mimic some of the vulnerability that's seen in humans by removing these protective factors in the mouse. But also, we could try to take some of these protective factors from the mouse and use them as kind of a therapeutic tool in humans. So this cross-species comparison is, I think, really important, both for making better models of a disease and for identifying therapeutic targets.
So, not that we want to do this, but there's some species that are very intelligent. Chimpanzees or gorillas, or is that a part of this cross species look too?
So it's really not possible, or it's probably not desirable to do research on great apes like
Yeah, they're
gorillas and
so
chimpanzees.
rare. We don't want to do that. Yeah.
Yeah, and they're so close to humans. But we can do some research on macaques that are a little bit further away from humans, and we can try to understand a little bit in the similar vein as with the comparison between mouse and humans, trying to understand what makes an entorhinal cortex layer 2 neuron in macaque compared to humans, and see if there are some protective factors in macaques. And I think that when we'll start to understand a little bit better the particularities of these neurons in different species, we'll be able to understand what type of impact evolution had on these neurons. And my guess is that at one point, there's probably a delicate balance to be found between making these neurons extremely plastic so that we can have a very great, very powerful memory that can store a lot of things and that can create a lot of memories in a very plastic way, but without risking of potentially having all your neurons that die at one point in your life. And so it's possible that in macaques, this balance is skewed a little bit more towards these neurons being a little bit more stable and a little bit more resistant. And in humans, they're a little bit more fragile. So it's possible that there's some aspect, and I'm talking as a complete ignorant of macaque memory, but it's possible that there are some specificities of memory formation in macaque that's very different from humans, in spite of the intelligence of these monkeys.
Yeah, that's interesting. So you talked about therapeutics. There's understanding that you're doing. Are you thinking about the potential therapeutics that might come from your research, and what might those be?
Yeah, very, very much so. And part of I feel like the idea of how to make more translatable discoveries in the lab that will be helpful for therapies in humans is kind of built in our research because we're trying to reshift the focus towards the neurons that are the first ones to be affected in humans. So we're trying to make a mouse model that would have pathological lesions in these neurons. We're trying to make these neurons in vitro. And so if we manage to get these better mouse models, if we manage to get these better in vitro platform, then we'll be able to test drugs in context that is a little bit more relevant to the human disease. Because so many mouse models of Alzheimer's have pathology everywhere in the brain when this is not the way it looks in humans. So many in vitro models of Alzheimer's, again, are made of neurons that don't really have much to do with the initial actors of the disease. And so it might be part of the reason why it's been so difficult in the field of Alzheimer's to have drugs that manage to be translated from the preclinical models into clinic, because probably the models that we were using preclinically were not quite as relevant as they should have been. And so our hope is that part of our research trying to make mouse models, better mouse model of Alzheimer's, are going to really provide the field with a better preclinical model in which we can be more confident that a drug that seems to have an effect for Alzheimer's or for some aspect of Alzheimer's is indeed going to make it in humans.
So when you say preclinical, is this early stage Alzheimer's where people are just, or explain that?
No, I meant more in a setting where you show that a drug is promising before you can then take it into a clinical trial. And before you take it in a clinical trial, you want to have a system in which you're showing that your drug is actually good at either, for example, wiping out amyloid plaques from the brain, preventing this protein tau from aggregating, preventing neurons from degenerating. But these demonstrations that are needed before you would take a drug into the clinic, they need to be done in a model that's relevant for the disease. And if they're not done in a model that's relevant for the disease, we're going to have over and over again drugs that seem to be very promising in the mouse and are actually not promising at all, but that are actually not working at all in humans. So I meant more like we need to have a better way of assessing if a drug is going to be efficient or not before we bring it to humans.
Which would we need new diagnostics then too? Because if this is the first place that the disease starts to manifest, that could go, kind of much younger than what we have typically seen, where when people go to the doctor and, it might be 20 years that they've had this disease. So do we need better diagnostics?
Absolutely, this is such a great point. So there's some studies that seem to indicate that by the moment when you have your first clinical symptoms of the disease, you already have more than 50% of these cells that have died. And so we obviously want to start treating people before there's already half of that neuronal population that's wiped out. So if we manage to find drugs that are preventing these neurons from malfunctioning, forming protein aggregates, dying, will need to treat people before they have clinical symptoms. And I actually find the diagnostic field in Alzheimer's one of the most exciting fields, where there's all of these new tests in plasma. So you don't need to do very complicated and very costly in vivo imaging in order to see the disease. We are directing ourselves towards the possibility to actually diagnose the disease before the symptoms from just taking blood. And so I think that this is going to be really, really instrumental for allowing therapies that are directed at these entorhinal cortex layer 2 neurons to function correctly. So we are definitely going to need very, very early diagnostic in order to be able to use these therapies efficiently. So definitely the necessity to find therapies for these cells, it's really important because we want to prevent these cells from dying. So if the cells are dead, we're not going to be able to, unless we're going the route that you were mentioning before, trying to make these cells in vitro and graft them into the brain. This is going to be really difficult. So we really need to act before the cells are dead. But the other aspects that I didn't talk about yet is that there's been a lot of speculations and actually a lot of very convincing evidence for the spread of toxic protein aggregates from one brain region to another. And so you would have toxic protein aggregates that would travel from one brain region to another using axons in particular. And there is some convincing evidence that the very first region where the toxic protein aggregates are coming from, it's entorhinal cortex layer 2 neurons. So to try to imagine what's going on in the brain of somebody with Alzheimer's, these neurons are the very first ones that start to malfunction, that forms these aggregates, and then they are sort of contaminating the rest of the brain with these aggregates. And so if we prevented these neurons from forming these aggregates in the first place, we would just, cut the disease before it can even spread to other regions of the brain. So it's probably going to be much easier to kind of curb the pathology when it's still very localized than when it's everywhere in the brain and every brain region is affected. So I just wanted to highlight the fact that this was another reason why I think it's really, really important to try to tackle the disease very early on. It's when it's still very localized to this region and hasn't spread to very wide brain areas.
Boy, that would be amazing. And then to have some kind of tests that you can take that maybe even be routine when you start in your 20s and 30s to be able to identify this. That would be incredible. We've talked a little bit about collaboration and, with some of the researchers that you're working with, but how important is it for academia and industry and any kind of regulatory agencies to really be working together to help kind of achieve that, that vision that you laid out here? And then not just collaborating, but what could they be doing to help accelerate it?
Yeah, that's a great question. So we don't have yet any partnership with the industry, but I'm hoping that some of the models that we're making, once we demonstrate their relevance for the disease, that they might interest people, and that they might be the basis for screens for drugs that could prevent degeneration. But I think it's really, really important because you don't have the same our small in the greater scheme of thing, academic labs can elaborate these new models, can test specific hypotheses, but we're never going to be able to have the same power to make a giant drug screening thousands and thousands and hundreds of thousands of compounds in order to find one that is actually helping our neurons survive. And so I think this is where the industry is uniquely poised for that. They have some robotics tools that we don't necessarily have very readily in academia in order to test all these libraries of compounds. And we always have, obviously there's all sorts of scales of labs in academia. You have small starting labs, but then like mine, and then you have huge labs. The labs where I was doing my postdoc in New York had 50 or 55 people in its heyday. So obviously, these two scales of labs don't have the same throughput. They don't have the same power to test many, many hypotheses. We have to be a little bit more selective in what questions we're asking, but definitely industry has a power that academic labs don't have.
How easy or difficult or just expand? I mean, I'm curious because the Karen Toffler Charitable Trust gives grants, and it gives grants to early researchers that are, kind of helping get their labs started and work on things that maybe other people aren't working on, kind of the mainstream. How difficult has it been for, and I don't want to put you on the spot, but funding's a big issue. And especially for, these startup labs that are smaller. How hard is that? And what do you need help from that community? Because there's family offices and there's all sorts of investors that would love to help you. They just probably don't know that you exist, but maybe expand that.
No, that's a great question. And I'm super grateful of the support that we received from the Karen Toffler Trust. And I think it makes such a huge difference for starting labs because in general, you have some hypotheses, you have some preliminary experiments that show that your hypotheses are actually interesting, that they're promising, but you don't have the huge power that very big labs have to demonstrate that they can lead huge projects. And so it's really important to have these grants from charities, from associations that are getting you started, that allow you to generate preliminary data, that allow you to show that you can do complicated experiments, that your research directions are promising, and then you can actually go to try to get federal money. And it is challenging to get federal money. For example, in the Alzheimer's field, which is one of the best endowed fields when it comes to biomedical research at the federal level, with anywhere between 15 and 25% of grants at each application cycle that gets funded. So that's a lot compared to the fields of saying anything but like epilepsy or Parkinson's or even cancer. And so we're very, very fortunate in the field of Alzheimer's to have this facility compared to other fields, but it's still only 15%, right? So that your project, you need to prove that your project is extremely competitive. You need to prove that your preliminary data is extremely convincing, and it's with starting grants like the grants from the Toffler Trust that can allow us to do these kind of things. It's really great to have this kind of two-gear system where we can get started with the money from charities, and then we have the federal money that allows us to kind of expand our projects and be more ambitious.
Yeah, that's been the probably one of the most surprising things to me, kind of delving into this the last 3 or 4 years, is that a small amount of money can allow for researchers to publish, and that these publications are so important. And so it's not the money necessarily to an outcome, to a therapeutic or a diagnostic or a cure. It's really this kind of lily pad of research that is a collection of publications that will lead to, that support that may create the breakthrough.
Exactly. And I think it's so hard to imagine that research is working like this for the general audience, because we'd love to have a research community that can, start working on a problem and then 5 years later there's a drug. But it doesn't work like this. We need these lily pad of discoveries. And definitely, I think that one thing that's great with grants like the Toffler Trust grants is that so it can allow starting researchers that are coming with fresh ideas that are not necessarily thinking exactly like the more established researchers of the field. And it can allow these young researchers to propose a brand new research direction that, we all hope is going to end up making a lot more people switch towards this new direction. And I think that this is very often what's happening, and especially in a field like Alzheimer's, where I have to admit that the therapeutic options that exist out there for Alzheimer's are not necessarily super encouraging. We still have to do so much progress in order to manage to find a disease-modifying drug. And yet, the field of Alzheimer's, it's not a new field. The disease was discovered more than 100 years ago. There has been very intense effort on the disease for decades now. And we know a lot. It's not like we've only done useless things for decades, but we know a lot about the disease, but we're still not there yet when it comes to finding good therapeutic targets for drugs that would really make a difference.
It's so complex. And the brain and the brain-body connection and what's going on. And that's why it's really important, I feel, to have a portfolio of people and effort, the more established, as well as just higher risk taking to just see and to test. And it's a smaller amount of money than these gigantic grants, but it allows for, just some preliminary kind of what if. Yeah.
Absolutely.
Yeah. So important, because we need it all, right? It's just, these diseases are so devastating and horrific that, we need that. So as you look out and, you've talked about your hopes and what you're focused on, but really, if you step out 5 or 10 years from now, what do you really hope to see, and that expectations that you're so passionately working towards?
Yeah, so I think that I'm hoping that the field And so I was going to say, I'm hoping that the field is going to understand that there's probably not going to be one therapeutic target, one direction that's going to be successful to treat the disease. But actually, I think that the field already knows that. But there is a little bit a sense in the field that whenever there's a new hypothesis, everybody starts to work on it. And I think it's really, really important for the field to keep in mind that we need these different research directions and that we might actually need, in order to treat the disease, we might actually need to hit different targets. We might need to prevent neuroinflammation, so prevent the local immune cells in the brain from doing damage to the brain. We might need to continue improving on the treatments that are targeted at amyloid plaques. We might need to try to more directly protect the cells that we're working on in the lab, the cells from the entorhinal cortex layer 2. So we might need all of these different interventions. We might also need some behavioral interventions, making sure that elderly people remain in a social environment and remain socially stimulated, that people do exercise. We might need some diet interventions. So I think it's important to keep in mind that all of these things are going to potentially be making a difference for people with Alzheimer's.
Yeah, and it just circles back to the diversity of all of us. We may need that, and it might be very, very personalized. I think we've hit a lot of different topics and different kind of angles to this. When would be a good time to circle back? I mean, with you, because these things take time, but is this like, do you like in a year, 2 years, will you have just 6 months? Will you have even more information to share? Give us some way.
I'm hoping that we'll have tons of information to share already in a year, but probably in a couple of years would be a more realistic estimate of when there's going to be brand new things to talk about with excitement.
Yeah, it's the timing clock of a researcher is very different than some industries. Yeah.
Unfortunately.
How would people, if they're interested in what you do or collaborating or just finding out more, how would people get a hold of you?
Sure, I would love to hear from people that are interested in continuing the conversation or collaborating. I think just shooting me an email at jproussa@bu.edu would be, I think, the best way to reach me.
Yeah, and people can always Google you, and there's a lot written in your publications that are out there too. So.
Mm-hmm. And we have a lab website that has more information about what we're doing. If you Google my name, you'll bump into the lab website very easily.
Well, Jean-Pierre, thank you so much. We don't get to talk as often as what I would like, but man, your energy is contagious, and your passion, and I love the focus. And I bet you it's a lot of fun to work in your lab. So thank you so much for this time.
Thank you so much, Deb, for having me. It's been just so great to interact with the entire Toffler Trust, and it's been great to talk with you about these topics that are very dear to me.
Wonderful. Well, till next time.
Till next time. Thank you.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.